Hello students, now we will start with a new topic that is about proteins. Proteins are synthesized by amino acids, means many amino acids are combined to give out proteins. These amino acids are defined as methane derivatives or substituted methanes. Why? Because the basic structure of amino acid has one carbon and many number of hydrogens, but among those hydrogens, some are replaced by different types of functional groups. For suppose we know that methane general formula is CH4. It is having central carbon and basing on the valency of carbon, it is having 4 hydrogens around it. Any 3 of these 4 hydrogens are replaced by different functional groups. In case of amino acid, those functional groups can be one amino group, one carboxylic group. It can be another methane or another hydrogen or another alcohol group. So we call such types of combinations as one amino acid. So we are telling, since we are replacing or substituting hydrogen with another different types of functional groups, we are telling the amino acids are substituted methanes. In universe or in nature, there are many number and many types of amino acids. But only out of those, 20 types of amino acids are involved in protein synthesis. For suppose, what are other types of amino acids that are not involved in protein synthesis? If you take one example, ornithine. Ornithine is also one of the amino acid only, but it is not involved in protein synthesis, but it is considered that it is used in stress reliever. It is used or involved in relieving stress. Next, in amino acids, basing on the number of amino groups or carboxylic groups, again they are classified into basic amino acids and acidic amino acids. Basing on number of amino groups and carboxylic groups, amino acids are classified into one, basic amino acids, second, acidic amino acids, third one, neutral amino acids. In case of basic amino acids, there are more amino groups. This we can remember. Here examples are arginine and glutamine we can take, or aspargine and glutamine we can take. Aspargine, glutamine. Now, in case of acidic amino acids, we can tell there are more number of carboxylic groups. Here we can take examples like aspartic acid. Next, glutamic acid. In case of neutral amino acids, what are they means? They have equal number of amino groups and equal number of carboxylic group when we compare amino and carboxylic group together. Means if one amino group is there, only one carboxylic group will be there. If two amino groups are there, again 2 carboxylic groups will be there. So under this, examples are glycine, serine, etc. So these are basic, acidic and neutral amino acids. Neutral amino acids have equal number of acidic and basic groups. Next we will see, basing on the requirement to animal body, what are the types of amino acids. Here amino acids, some types are already synthesized in animal body. Those are considered as non-essential because they are already synthesized in the animal body. So animal no need to take the required amino acids in the form of diet. But some amino acids are not synthesized in animal body. So the animal, to fulfill its metabolic activities, it has to intake some types of amino acids in the form of diet. So they are called as essential amino acids. Basing on requirement of amino acids to animal body, amino acids are again classified into first, essential amino acids, second, non-essential amino acids. What are essential amino acids? We have equal number of essential and non-essential that are listed here. Under this, we can write valine, histidine, tryptophan, isoleucine, leucine, lysine, methionine, phenylalanine, okay, next threonine, arginine. So totally 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So here we are seeing 10 number of essential amino acids. Already I said you only 20 number of amino acids are involved in protein synthesis. So we have listed out of the 20, 10 here. Means these are not synthesized in animal body. So animal has to take all these types of amino acids in the form of diet. Coming to list of non-essential amino acids. What are they? Glutamic acid. Glutamic acid. Next is glutamine. Glutamine. Next one, aspartic acid. Aspargine. Glycine. Serine. Tyrosine. Proline. Next, alanine. So we have listed 1, 2, 3, 4, 5, 6, 7, 8, 9. One more thing is there, serine, glycine, proline, alanine I have written. Next, aspartic acid, cysteine we did not mention. Cysteine. Cysteine is sulfur containing amino acid. So these are non-essential amino acids and these are essential amino acids. Now, out of these essential amino acids, phenylalanine, tyrosine, and tryptophan are considered as. So phenylalanine and tryptophan are essential amino acids as we have mentioned here and here. But tyrosine we have mentioned here under non-essential because already it is synthesized in animal body. So tyrosine, these three are considered as aromatic amino acids. Aromatic amino acids. So we label also this one simply as what are AAA amino acids. AAA types of amino acids means aromatic amino acids. So the listed are 3 in number: phenylalanine, tyrosine, and tryptophan. One more function we can see. There are some types of amino acids basing on the requirement in the animal lifetime. These are also graded into semi-essential amino acids. Semi-essential are amino acids. What are these? These are required only in few stages of animal lifetime, means required only during animal growth, and also during lactation period in females. Here examples are histidine and arginine. Histidine and arginine. Histidine already, I think I told you, histidine is used during the growth. At the same time, simultaneously, it is helpful in repairing of the damaged tissues. And cysteine is sulfur containing amino acid. These three contains aromatic side chains in their structures: phenylalanine, tyrosine, and tryptophan. And these are essential. If you see the number also 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So here also I have listed 10 number of non-essential amino acids. So 10 plus 10, 20 number of amino acids list is given here. In the same way, in case of proteins which are framed out of amino acids, we see peptide bonds. So, peptide bond formation. Now, we will see the basic structure of the amino acid. As I told you, methane derivatives we have said. So, we can give the first type of structure before complete successful formation of amino acid like this. Methane structure like this. From this, we are replacing or substituting some functional groups: NH2, COOH, R group, and hydrogen. This is the structure of amino acid. One R group we did not mention any. So, if that R group is again replaced with any hydrogen, if the R group is replaced with hydrogen, then the structure will be like this. We can give a name for this amino acid as glycine. Glycine is considered as the basic amino acid structure or basic amino acid. Now, if the R group is replaced by one methane group, then we can name this amino acid as alanine or alanine. Again, another amino acid. If it is R group is replaced by any alcohol group, then there will be another name for that amino acid. We can name out as serine. Let's take any one of these amino acids and show how the peptide bond is formed. Since this is easy to understand and easy to take, let's take two glycines and give out the peptide bond appearance. You can simply write R group. Like this. Now it has to bind with another R group containing structure, amino acid, next COOH, like this. But I told you we will take glycines as example and show the peptide bond formation. So this amino acid is glycine. This amino acid is glycine. So we'll write on the top. Now we are fusing two glycines to give out a new structure. So we can write this one as like this. One hydrogen of amino group of one amino acid and hydroxyl group of other amino acid, both are removed in the form of water before formation of peptide bond, and here forms the peptide bond in between two amino acids. If the peptide bond is formed, how can we represent that? C, H, H, now H, N, O, C, again amino group, again hydrogen. This combination of hydrogen, nitrogen, oxygen, carbon is called as peptide bond. Peptide bond is also formed by the dehydration between two amino acids. Same in the carbohydrates also we have discussed. Glycosidic bond is formed by dehydration between two monomeric carbohydrates or monomers or monosaccharides. There also dehydration happened. Here also dehydration happened. But in case of carbohydrates, the bond is glycosidic bond. Here the bond is peptide bond. In living organisms body, these amino acids are also existed in ionic form. We give a term for them as Zwitter ions. So, amino acids in ionic form we can write as one R group, one hydrogen, next NH3 plus COO minus. So this we are calling it as Zwitter ionic form. Means later it can form and complete its valencies and addition of any of the groups like hydrogens, hydroxyls, etc. And it can further involve in the metabolic activities. So this is about peptide bond and its formation. Next we will see term is given by Berzelius. But detailed study of proteins was done by Mulder. Detailed study of proteins was given by Mulder. Now, these proteins are also termed as polypeptides. Polypeptides. Now, if the protein is made up of one polypeptide, if means one polypeptide is behaving as a complete protein. Protein made of one polypeptide. Then it is termed as monomeric proteins. Then it is termed as monomeric proteins. Examples we can take myoglobin protein, which is seen in case of animal histology topic. Another example, lysozyme, which we study this one existing as in saliva of human body to kill the bacteria that is entered in the form of food. Next. So this is about monomeric. If two or more polypeptides behave as one complete protein, then those are called as multimeric proteins. There we can take example as insulin. Behave as one complete protein. Called as multimeric proteins. Your examples we can take as insulin. A functional insulin, it is having two number of polypeptides. One is A polypeptide and B polypeptide. That only we call it as insulin is made up of one alpha chain and one beta chain. So, if you write the structure like this, which are bounded by disulfide bonds, this is made up of A polypeptide chain and this is of B polypeptide, which are both connected by disulfide bridges. So, insulin made of one alpha chain means polypeptide and one beta chain means B polypeptide has 4 stages to form a structural or complete protein. First one is primary structure. Mostly, primary structure of the protein is considered as non-functional and the shape is termed as linear shape because it is not mechanically fit, it is non-functional and there are no any turns to give the mechanical support to the protein. So, these are made of only amino acids and peptide bonds. Shape is linear and considered as non-functional form of protein. Non-functional in nature. This primary structure of protein is considered as non-functional, means it is not involving in any function. Here we see only one type of bond, that is peptide bond, that is formed between amino acids. But exception is there. Exception is insulin. So, insulin is nothing but the primary structure of the protein, but still it is functional. It is responsible to decrease the glucose levels in the blood of human beings or animal body. Insulin is functional in its polypeptide or primary structure form. Next one, secondary structure of protein. Now, the shape changes from linear to helical form. Shape is helical in nature and is again considered as non-functional, but still here there are some some exceptions here, just like that of insulin in primary structure, and is also considered to have 3D form or 3D structure due to turns or helical arrangement in structure. What is this? We'll see now. Here we are seeing arrangement of the structure as like this only, one complete linear form of the shape in primary structure. But coming to secondary structure, there will be turns or coils are seen. Means the linear form, now it has framed out some of the coils like this. These coils are maintained, means maintaining their structure with the help of hydrogen bonds like this. Like this. Means how many types of bonds are there now? One is peptide bond between the amino acids. So if we take two amino acids, one amino acid here, one amino acid here, which are formed into coil-like structures, this one we can call it as peptide bond. Another type of bond is hydrogen bond. Now, this secondary structure also exists in beta plated form. Means two number of alpha helical structured proteins are binded one with each other and are framing into secondary structure of the protein. So we are calling one as alpha helix form. Now here two number of alpha helices are involved. So this is called as beta plated structure or sheath. Two alpha helices are bonded by hydrogen bonds. How? So take amino acid, another amino acid, another amino acid, another amino acid. This we can call, this is nothing but peptide bond. And among the turns also we have hydrogen bonds already existed like this. Like this. But to have a stable structure like beta plated sheath, between two alpha helices also there are hydrogen bonds. Like this. So the structure is maintained very unstable in nature. There is no any deviations in the structure of the beta plated sheath when two alpha helices are fitted with the help of hydrogen bonds. Here we can take two examples. One is keratin protein. Second is silk fiber we can take, which is obtained from silk moth. Silk fiber. Since we are studying the existence of one alpha helix form in secondary structure, beta plated form in secondary structure, here keratin protein is having beta plated sheath, but still only one of the beta plated sheet, that is one alpha helix form, is functional in case of keratin protein, and both the alpha helices in the form of beta plated sheath are functional in case of silk fiber. That I will write here. Keratin protein, which is seen in our hair and nails, etc. Keratin has beta plated sheath structure, but still only one alpha helix form behaves as complete protein. Next one, silk fiber, which is obtained from silk moth. Both alpha helix forms of beta plated sheath combinedly behaves as complete protein. Now, we can also call like this secondary structure of the protein is giving us one complete 3D structure of arrangement. Next one is tertiary structure of protein. I am erasing this secondary structure. We have to remember that in case of primary structure, there is only peptide bond between the amino acids. In case of secondary structure, there are two different types of bonds. One is peptide bond and another is hydrogen bond. Tertiary structure of protein. Here we see many different types of bonds and also perfectly framed out 3D shape of the protein. 3D shape of protein is due to many foldings within the strand or within the chain. Number of polypeptides involved are 3 polypeptides. Frame out into tertiary structure of the protein. So I am giving out the structure: one alpha helix form, another alpha helix form, another alpha helix form. The structure is arranged like this. Here we have to remember, if one end is having amino group, other end has carboxylic group. Other end has amino, other carboxylic group, other amino, other carboxylic group. The structure is arranged like this because these gaps are called as active sites. At this tertiary structure of the protein, some proteins are separated in the form of enzymes. Some proteins are limited only up to that structure, and some are behaving as enzymes, some are behaving as hormones, etc. Now, so here also another active site. Okay. Some tertiary forms are limited in their structure to behave as enzymes, hormones, etc. But some will be extended to further structure, that is quaternary structure. Now we will see why, means what is the importance of existence of one amino group and one carboxylic group in tertiary form. Why means we know that during enzyme and substrate reactions, enzyme binds the substrates at its active site. Since we are calling this shape as this place as active site of the enzyme, so one substrate has to come and bind here. So it will form a bond with OH group here and one amino group with here. So this one we can consider as the substrate, so that later it gives out products. At the same tertiary structure, means 3D structure of the protein, most of the tertiary structures are considered to be active in form. If we extract the structure like this, means a little bit of part I have drawn here. In primary structure we have seen peptide bonds only. In secondary structure we have seen peptide bonds plus hydrogen bonds, means I am mentioning here hydrogen bonds like this. Between the amino acids there are peptide bonds. Outside the turn or outside the coil, this is turn or coil. Outside the turn there are some special type of other bonds also called as hydrophobic bonds. Within the coil or inside the turn there are ionic bonds and disulfide bridges or bonds. If we count what are the types of bonds in tertiary structure, we see hydrogen bonds are existed, peptide bonds are existed, hydrophobic bonds are present outside the turn, inside the turn ionic bonds and disulfide bonds. So these are present inside coil or helic form or inside the turn. So here we cannot specifically take one or two types of names, but simply we can write mostly enzymes. But there are some types of tertiary structure of proteins which are specially functional, and they enter into the structural framing out quaternary structure of proteins. In the quaternary structure of proteins, four or more than four number of polypeptides are involving in giving out one complete functional proteins. So here 4 or more than 4 polypeptides or alpha helices chains are formed. We can take alpha helix forms are formed into complete functional protein. Examples we can take as hemoglobin molecule, because in case of hemoglobin molecule we see it is made of 2 alpha helices and 2 beta chains. 2 alpha chains and 2 beta chains. Now, the subunit means here the complete protein structure depends upon the subunits of these alpha helices or beta form of chains. Along with this, we can also take another example, that is aquaporins. Aquaproteins are the carrier proteins that are supported in transportation of water molecule between the plasma membrane into the cytoplasm or outside the cell body. So aquaproteins are carrier proteins that transport water molecule. Water. Now, 8 aquaporins are termed as or are called as one water channel. This is also your NCERT MCQ, direct point from NCERT. So, this is the structure of proteins. We have seen the primary structure, which is non-functional, but exception is insulin. Secondary structure, still it is non-functional, but exceptions are keratin and silk fiber. Keratin is having 2 polypeptide chains, but still only one is functional there. Means, okay. Next is silkworm or silk fiber, which is obtained from silk moths. There, 2 alpha helices of the beta plated sheet are functional. Next, coming to the bonds. In primary structure, we see only one peptide bond. In tertiary structure, we see 2 bonds, that is peptide and hydrogen. In tertiary structure, we see peptide bond, hydrogen bond, hydrophobic bond, disulfide bond and ionic bond. 5 different types of bonds in tertiary structure. Enzymes are nothing but they are limited only up to the tertiary structure of the protein synthesis, which are having active sites. Next and the last one is quaternary structure of the protein, in which completely they are responsible for structure or complete molecule formation, just like aquaporins and hemoglobin, etc. Whatever the aquaporins or hemoglobin may be, their structure depends upon the subunits like number of alpha helices and their arrangement, etc. So this is about protein structure. Basing on composition of protein. Again, proteins are of different types. I am not telling here their number and all that. We are seeing the types. Proteins are classified into first simple proteins, second one conjugated proteins. Simple proteins means only amino acids giving out into proteins. Only proteins will be included in that complete structure. For example, if you take albumin, albumin is one of the functional protein that is having only a protein type of biomolecules. Carbohydrates, all these are not involved in that. So, if proteins are made of only amino acids, then they are called as simple proteins. So, example is albumin. Next, globulin. Next, Another type of amino proteins are conjugated, means protein part plus non-protein part is included in conjugated proteins. So proteins, we know nothing but combinations of amino acids, but this non-protein, it can be a carbohydrate, or it can be a nucleic acid, or it can be a nitrogen base, etc. Okay, it can be anything. So basing on the combination of non-protein part with that of protein part, the names are different. Now we will write few names of that. That is protein plus carbohydrates are termed as glycoproteins. Mostly we read glycoproteins are existed while drawing the plasma membrane structure. Next, protein plus nucleic acids like DNA fragments or RNA fragments. We call it as nucleoproteins. Next, protein plus lipids we call as lipoproteins. Next, protein plus phosphate molecule or phosphate ion. These are called as phosphoproteins. This can be seen in case of plasma membrane. This can be seen in genetic material. This can be seen again in plasma membrane or inside the cytoplasm. Then what about these? Phosphoproteins are seen in casein. Casein is a milk protein. Proteins also bind with some of the coloring pigments like heme, then forms into hemoglobin. Globin part is a protein. Protein plus coloring pigment. So, example, heme. So, basically we can give a term like chromoprotein. The same chromoprotein, if it is having heme, then we are calling as hemoglobin, because this protein can be globular protein. Next, proteins basing on different functions. First one, structural proteins. Examples we can take as collagen. Collagen is considered as the abundant protein in animal kingdom. In plant kingdom, what is the abundant protein? Nothing but enzymatic form, that is rubisco. Collagen, elastin, etc. Second, storage protein. Here we can give example like albumin. Third one, enzyme forms. Fourth one, hormones. These enzymes and hormones are framed out from the tertiary structure of protein synthesis. When protein is getting synthesized, at the tertiary structure, these are deviated into enzymes and hormones. Next, proteins in form of immunoglobulins. In form of immunoglobulins. They provide immunity. They provide immunity. Next. Proteins are also involved in blood clotting process in the form of fibrinogen and prothrombin protein. Proteins also involved in blood clotting. So what are proteins that are involved in blood clotting? Example, fibrinogen and prothrombin. Next, proteins also support in transportation of different solutes or different solvents from outer to inner part of the cell or from inner to outer part of the cell. We can take best examples as porins, and for water transportation, aquaporins. Some types of proteins also use energy and they transfer the solutes or the substances. Those are called as pumps. So here we can take carrier proteins like porins. Porins are considered to be existed in outermost layer of mitochondria and outermost layer of plastid. Pumps. Pumps are considered to be utilizing ATP and doing the transportation. These are also carrier proteins. But porins will not use ATP. Those are considered as pure facilitated diffusing carrier proteins. Next, another example we can take porins, pumps, and also aquaporins. Aquaporins transport water molecules. 8 number of aquaporins are called as one water channel. Next, proteins are also used in contractions of muscles, and there the types of proteins are nothing but actin and myosin. Proteins in form of actin and myosin helps in contraction and relaxation of muscles. Of animal muscles we can say. Next, hemoglobin is used. I think we have not mentioned hemoglobin. So globular protein in hemoglobin, it is supporting in transportation of in the complete hemoglobin form. It is helping in transportation of oxygen to all over the body through blood. So oxygen transporter in form of hemoglobin. Hemoglobin I am writing as capital H, small b. Next. So there are some types of proteins which support the neighboring proteins or other proteins to fold in a proper manner and give out a complete structure of the organism. Such types of proteins which are involved in proper folding of other types of proteins are called as chaperones. Some types of proteins help other proteins to fold in proper manner to achieve functions, means functional capacity. Example for such proteins which are supporting other proteins to either form into 3D forms, etc. Those are called as chaperones. Now, according to NCERT, another example is left over, that is glutamin 4, that supports in transportation of glucose among the cells. So, GLUV, they have given like this, and Roman letters 4, GL, okay, UT I think, okay. Glutamin 4 enables glucose to be transported into the cell. Some types of amino acids like trypsin in the form of enzymes, some types of amino acids in the form of receptors also allow or support in transportation of different hormones, different types of solute particles into the cell and out of the cell. So this is about proteins, their peptide bond formation, different types of amino acids, different functions of proteins, and we also have discussed about primary and secondary metabolites, etc. So this is about complete proteins in biomolecules chapter in biology, that is required for NEET purpose point.